Hi and welcome to this video, which is an update on the trials we've been conducting here at Plato Bio on our product Stubble Digester. Stubble Digester was a product that we developed to solve the problem of crop residues on arable fields. Crop residues are a real problem in many farms worldwide, and that's because the stubble that remains after a crop has been harvested interferes with the next crop that is sown. That's physically in interfering with the actual sowing process and the machinery, and also nutritionally, because that crop residue, when it's being broken down, is being taken over by fungi that absorb nitrogen from that residue and from the soil that would otherwise be returned to the plant. Other nutrients are important and are locked up as well in that process. So the quicker you can break down that crop residue, the better. And that is what Stubble Digester does. So in 2019, we set out to get more data on this process using this product. So we had a two-pronged attack. The first was to conduct field trials with Stubble Digester with our partner in the UK, Agrovista. So a large sample of Stubble Digester has gone out into fields to look at the breakdown of the stubble in a real-world situation with this partner. The second prong of this attack was to look at it more closely on a lab scale. So we took subsamples of stubble from wheat and looked at the decomposition rate in the laboratory. We also looked at thatch, which is the dead leaves from grass, because this is another crop residue problem in sports turf and amenity turf situations. So over a number of different timescales, we looked at the breakdown of these residues, and then we looked at that compared to a water control and a nitrogen treatment, which is an industry standard treatment for the breakdown of crop residues. And we were really impressed with the effects of Stubble Digester. Within 7 days on grass turf thatch, we found a breakdown of the residue, and that residue had lost 30% of its dry weight. We saw similar effects in wheat crop residue over a longer time period, but what was most exciting about the wheat breakdown was that we saw a proliferation of the saprotrophic fungi that break down the residue. These are the fungi that live off dead and decaying plant matter. There was so much fungal breakdown on the wheat residue treated with Stubble Digester that it actually grew toadstools. So we could identify the species as well. So we could see that these were common ink camp fungi. These results mirrored the results we got in 2018, where we applied Stubble Digester to a normal grass turf lawn and found that we had a proliferation of saprotrophic fungi, and we could identify the species again, and it was clear that one side, the treated side, there were loads of toadstools and there were none on the other side. Another experiment we conducted was to look at the tensile strength of the stubble that had been treated. So again, it's a wheat treatment here, and with a water treatment alone, there was some breakdown of the stubble. With the Stubble Digester, the stubble that was treated was a lot easier to break. So physically, in our hands, when you tried to break the stubble, it was very easy to break down. And then the urea ammonium nitrate treated product was actually very, very difficult to break down. And that's because the nitrogen and the ammonia had actually sterilized the stubble. So a high treatment of nitrogen was actually counterproductive to stubble breakdown. So this was a really interesting finding. Other options you might have seen for breakdown of stubble include molasses. This supplies a source of energy to the saprotrophic fungi, which is a good way to break down the stubble. However, the advantage of Stubble Digester is, as well as providing energy, it provides the right form of energy for the saprotrophic fungi to drive the Krebs cycle, and it also provides nutrients, the elemental nutrients that are required by the fungi. So there are certain nutrients that these fungi require that wouldn't be in a conventional fertilizer, and the ratios would be slightly different. So we target them just for the saprotrophic fungi. So this is an advantage over molasses. Another alternative to Stubble Digester would be bacterial inoculants. However, at Plato Bio, we have taken the approach that there are already billions and trillions of bacteria in the soil, and adding what is relatively a few more is not actually going to help that. Because what you want to do when you're breaking down stubble is to target the breakdown of lignin. Lignin is the rate limiting step. The breakdown of lignin is the rate limiting step when it comes to decomposition of stubble. And the lignin forms the brown material in the stubble. Without lignin, stubble would be very easy to break down, and it would have strength, but it would not have any rigidity. It would be like my cotton shirt. Add more lignin, it becomes more like a wooden table. So it's the lignin that's actually giving it the physical strength and the problems that you see later on when it comes to sowing the next crop. So lignin is a phenolic compound that doesn't provide much energy. So when we provide the energy and the nutrients to the saprotrophic fungi, we can break it down a lot more easier. So that's the basis behind Stubble Digester. The trials that we've been conducting this year have supported that. We've got real-world situation data coming in from our partners in AgriVista. If you'd like any more information on Stubble Digester or any of the other products in our portfolio, please get in contact with Plata Bio or with our partners AgriVista if you're in the UK and in the arable sector. So if you've got any questions, please do get in contact. Thanks very much for watching. Goodbye.